Hello everyone. InformationBoxTicket Lifestyles brings you Virology, Human Papillomavirus HPV. But before that, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. Human Papillomavirus HPV. More than 90% of cervical cancer cases are found to have the human papillomavirus, which is also the most frequent infection linked to human malignancies. The HPV virus has more than 100 subtypes. Multiple epithelial lesions and malignancies, primarily on cutaneous and mucosal surfaces, are its major associations. It is the most widespread STD in the world and has a major negative influence on a person's social life. Genital and cutaneous warts are caused by low-risk HPVs, whereas oropharyngeal malignancies and anogenital cancers such as cervical, anal, vulvar, vaginal, and penile cancers are caused by high-risk HPVs. Although HPV does not directly cause cancer, it can be brought on by a number of other factors, including smoking, exposure to UV rays and other carcinogens, a lack of folate, immunosuppression, and pregnancy. Let's dive into the table of content of this chapter. We will learn structure of the human papillomavirus, genome structure, epidemiology, transmission, replication, including an early phase of viral replication and late phase of viral replication, pathogenesis, clinical manifestations, mucosal human papilloma infections, cutaneous human papillomavirus infections, its diagnosis, treatment, vaccination, and lastly, prevention and control. Let's get started. First, we will look at the structure of human papillomavirus. As you can see with this diagram, the papillomaviruses have an icosahedral structure, are tiny, non-enveloped, and measure 52 to 55 nanometer in diameter. A single double-stranded DNA molecule is contained within a protein capsid made up of 72 pentameric capsomers. Two structural proteins, including the viral protein L1 late 1 and L2 late 2, make up the capsid. At 55 kDa, the L1 protein accounts for 80% of viral proteins. As the size of the L2 is 70 kDa, the viral encoding of the L1 and L2 protein is the same. A variation number of L2 molecules, which are partially exposed on the surface of viron, are present in the viral coat together with 360 molecules of L1 protein, which are organized into 72 capsomers. In mammals and non-mammals, the expression of L1 alone or in combination results in virus-like particles. Viron density in cesium chloride is 1.34 gram per ml, while the sedimentation coefficient is 300. Kindly don't forget to show your support by subscribing to my channel. Genome structure of human papillomavirus. With the help of this diagram, you can see approximately 8 ORFs are transcribed into single double-stranded DNA molecule with a genome size of 800 base pair in the genomes of all human papillomavirus strains. The early region E, the late region L, and a sizable non-coding portion known as the long control region can all be classified as functional areas of the ORF, LCR. The L region codes for structural proteins, vital for viral assembly. The E region codes for proteins, E1 to E7, required for replication, and the LCR includes the cis elements required for viral DNA replication and transcription. The papillomaviruses varies in size and number of ORFs, but they all include well-conserved core genes for replication E1 and E2, and packaging L1 and L2, as well as very diverse genes for cell entrance, immunological infiltration, and viral release E6, E7, E5, and E4. Kindly don't forget to like this video, subscribe to my channel, and press the bell icon button. Epidemiology. The human papillomavirus has more than 180 known subtypes. Verruca vulgaris and Verruca platarius, which are cutaneous warts on the hands and feet, are brought by HPV subtypes 1, 2, 4, 24, and 57. These are transmitted by coming into touch with skin that has an epidermal damage or by infected insects. Dialoma acuminatum and human papillomavirus subtypes 6 and 11 are frequently linked to anogenital warts. They are known as low-risk HPV, and both juvenile and adult recurrent respiratory papillomatosis have been linked to them. Precancerous and cancerous regions of the cervix, male and female anogenital regions, and oropharyngeal regions are most frequently caused by subtypes 16 and 18. The development of cervical cancer is linked to the high-risk HPV subtypes 31, 33, 35, 45, 52, and 58. Despite being regarded as sexually transmitted diseases, both low-risk and high-risk human papillomavirus can spread through other types of intimate contact. According to recent research, 39.9% of women and 45.2% of men in people aged 18 to 59 have human papillomavirus, according to the Centers for Disease Control and Prevention. The most prevalent sexually transmitted infection in both the United States and the rest of the globe is the genital human papillomavirus infection. Now let's learn the transmission of human papillomavirus. Various HPV subtypes have an impact on the vaginal region, throat, and mouth. Despite being a prevalent STD, it has a potential to remain asymptomatic and go away on its own. The virus does not need to be penetrated during intercourse to propagate. It may transmit via skin-to-skin contact and the genital region and through fluid of the body. Among the risk factors for becoming infected with HPV are sexual activity, the first experience, and the total number of partners. Smoking. Oral contraceptives are used for more than 5 years. Consume betel nuts. Radiation and UV light exposure. Kindly don't forget to like this video and subscribe to my channel. Replication of human papillomavirus. Through microscopic tears, the HPV penetrates the cells. The single-layered scamovious cellular interface between the endo and ectocervix is where HRHPV, a form of HPV that causes cervical and other cancers, may enter the cell. The basal layer cell surface or the basement membrane includes cellular receptors such as heparin sulfate proteoglycan, which the HPV L1 capsid protein interacts to. Endocytosis, which shares the greatest similarities with the micropinocytosis, is how human papillomavirus enters the cell. The virus passes via the trans-Golgi apparatus and membrane-bound cytoplasmic elements. The viral episomal genome is subsequently transferred into the nucleus through nuclear pores or by the nuclear membrane rupturing during mitosis with a tubule-mediated route. The following steps of viral replication are early phase of viral replication and late phase of viral replication. Early phase of viral replication. Early transcription starts as soon as the nuclear entrance occurs. The E2 engages the cellular DNA replication machinery by binding to the E1, which then binds as a dimer of hexamers to the origin of replication site. In the initial replication, the HPV genome produces 50 to 100 episomal copies per nucleus. Late stage of viral replication. Viron development and viral DNA replication are both involved. Viral E1 and E2 proteins must express themselves more strongly throughout this phase. It is distinguished by the viral main late promoters activation, which is located in the E7 gene area and leads to the enhanced expression of E1 and E2 as well as E4 and E5. Thousands of offspring viral genomes are produced at the late stage of viral replication, which most likely utilizes the rolling circle process for DNA replication. After viral genome amplification and capsid protein synthesis, the viron is formed in the nucleus. The superficial keratinocytes are the site of viral maturation, which ultimately results in the release of the infectious virons. Kindly show your support to this channel by liking my video and subscribing to my channel. Pathogenesis of human papillomavirus. With the help of this diagram, you will understand the papillomavirus pathogenesis. Despite their high incident rates, HPV infections typically go away on their own within a year or two. The continued infection has a significant role in the emergence of cervical and other malignancies. Depending on the HPV type, HPVs cause vaginal and malignant lesions in the cutaneous and mucosomal epithelial cells. Because the host immune response mechanisms are so important in regulating HPV infection, a immunocompromised person has higher prevalence rates. The virus does not lyse the target cells after infection or generate viremia, which limits the exposure of viral antigen and helps the virus elude the host immune response. The MHC1 gene can be downregulated by HPV, and it can also interfere with the oncoproteins E5, E6, and E7 mediated interferons pathway. The Golgi apparatus transport HLA molecules and intracellular viral antigenic peptide complexes to the cell surface, where cytotoxic CD8 plus T lymphocytes detect them. The infected cells then undergo apoptosis as a result of activated cytotoxic T lymphocyte CTLs. The cytokine secreted by T helper cells promote proliferation, maintain cytotoxic lymphocytes, and activate B cells to promote antibodies. Clinical manifestation of human papillomavirus. The clinical symptoms of HPV include mucosomal human papilloma infection and cutaneous human papillomavirus infection. Mucosomal human papillomavirus infection has condyloma acuminatum, present as papules, nodules, or soft feriform pinkish siculi or pediculated growth in the genital area. Focal epithelia hyperplasia, disease of the oral mucosa, more common in children and women. Cervical neoplasia and cervical cancer. Other anogenital cancer including those of vulva, vagina, penis, and anus. Head and neck cancer. Cutaneous human papillomavirus infections. Common warts, plantar warts, flat wart, filiform warts, pigmented warts, epidermal cysts, skin cancer, which is Bowen's disease. How human papillomavirus is diagnosed. Human papillomavirus cannot be detected using conventional techniques like electron microscopy, cell culture, or certain immunological tests. Among the crucial techniques for its detection are whorls or condyloma detection. By analyzing the pigmentation, induration, ulceration, and other characteristics of whorls, one may visually check for genital or oral whorls, and a biopsy can confirm this. Colposcopy and acetic acid test. Following the administration of acetic acid solution and collection of samples from lesions suspected of being neoplastic, a colposcopy is performed to examine the cervix, vagina, and occasionally the vulva. The degree of aceto-white lesions, surface contour, mosaic pattern, and punctuations are used to score the colposcopic results. Based on anomalies of these parameters, the severity of lesions is deduced. Biopsy. Colposcopic results that demonstrate high abnormalities, low-grade alterations with severe dichrysosis, or a lesion that extends to canal need excisional biopsy. Genital warts are characterized by the presence of chyliocytes, meteosecmosis cells with a distinct perinuclear zone. DNA techniques. HPV detection was first carried out using direct probe hybridization techniques including dot plotting and Southern blotting. However, they needed a lot of DNA in the clinical samples, were time-consuming, labor-intensive, and had limited sensitivity. The two primary methods for routine viral detection are as follows. Hybrid capture HPV DNA test 2 HC2. The FDA has given the HC2 with a PAP test, it's okay. It can identify HPV as little as 1 pg per ml. Although it is unable to identify the precise HPV type, it can distinguish between the low-risk genotype group 6, 11, 42, 43, 44 and the high-risk genotype group 16, 18, 31, 33, 35, 39, 45, 51, 52, 56, 58, 59 and 68. Next is polymerase chain reaction. The MPV sequences found in the biological sample are amplified specifically. The next is Pap smear or Pap test. In the beginning it was explained by Paplinocoal and Trott. It makes use of the scrapings that are mounted on a glass slide from scamocolonal junction. This technique may be used to identify malignant and pre-malignant alterations as well as viral diseases like HPV and herpes virus. For cytological analysis, scrapings from the upper lateral region of the vaginal wall can be collected. Next, treatment of HPV. There are several ways to treat cutaneous warts, including surgery, cryotherapy, irritating or immunomodulating drugs, and laser excision. Podophyllin rhizins, podophyllitoxin, organic acid like salicylic acid, trichloroacetic acid and bichloroacetic acid, and cytostatic drugs like bleomycin, cidofovir, and 5-fluorouracil are a few examples of pharmaceuticals used to treat mucosomal and cutaneous warts. In immunocompetent people, organophenile and anogenital warts can both be treated. For the growth of HPV-related cancer, resection, chemotherapy, and/or radiation may be necessary. Vaccination of human papillomavirus. Currently licensed and available in the US is a 9-valent recombinant protein subunit HPV vaccination. The L1 major capsid protein is the main antigen utilized for HPV vaccination. Recombinant technology is used to create the L1 protein which self-assembles into particles that resemble viruses. It is intramuscular to deliver the vaccination. Routine immunization is not permitted for persons over the age of 45, but it is advised for both genders at age 11 or 12. Lastly, prevention and control. Although most sexually active persons eventually get HPV, there are few steps that may be taken to lessen the risk. The following are a few of preventive measures. At 11 to 12 years old, children should have their first vaccine. Vaccination is advised up to age of 26. However, if given after the commencement of sexual activity, the vaccine's effectiveness is reduced. Women with a history of intravasive cervical cancer, high-grade CIN, or endocarcinoma ensued should get an annual vaginal cough pap test. The incidence of anal cancer is PWH can be decreased in HIV seropositive individuals by screening for lesions and treating precancerous lesions. And that's it for today. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. Thank you so much for watching.